We having an official opening.
Yes, no.
You talk first.
You talk
or
first.
I talk first.
Welcome, Surat. You're welcome.
Okay. Official opening on behalf of Chulalongkorn University. I would like to welcome you all to this very important meeting on the ethics of human gene editing. I have been the director of this small Center of Ethics of Science and Technology here at Chulalongkorn for many years. We are a very small center, but our work is kind of wide-ranging because we have a lot of legworks. And one of our fellows who has been working with us for many years is, of course, Beryl. Beryl told me that he put me up giving an academic talk today, but I have not prepared anything. So maybe I thought it is going to be on Friday. So I have something to share with you on this topic. So I would like to be able to present, but hopefully not today. And if you have any problems, logistical problems, questions, feel free to ask me or Ananya or Daryl. We are working on the Wi-Fi problem, and I think the plan is for us to have our lunch there in the foyer, just outside of the room. Tomorrow, this room unfortunately has been booked, so we have to move to the room 706 over there. It's a bit smaller, but we don't have too many of us, so I think it will work. And starting 4 p.m. today and tomorrow and on Friday, we have to move to the other room 401/18, which is in the next building. There are two tall buildings, so we are going to move to the other one. We can have coffee break first, and then we move. So there's a break in between. So it's not much of a disruption. So welcome you to Bangkok. Another unfortunate incident is that we look out and you don't see much. Usually it's very clear blue sky, you can see far, but we are having this strange problem. We have not had this before. This is the first time we experience this. It's called smog or whatever. We call them PM 2.5. It's about very small particles, 2.5 micron across or something. And we hope that it will go away soon. So bear with us. So this is official opening, and let us to start having a good 4 days of academic presentation and discussion. Thank you.
Thank you, Surat. And I think it's nice to do self-introductions to each person, so I will start with Ryan on here.
Good morning to everyone. I am Christopher Ryan Magolok. I teach at the Philosophy Department in Ateneo de Davao University in the Philippines. It is my second time here at Chulalongkorn University.
Good morning. I'm Rogelio Bayod. I'm working at the Student Affairs Office of Curiaso College in Digos, this southern part of Mindanao. It's my first time in Chulalongkorn University.
Good morning, everybody. My first time in Chulalongkorn. Try my best, but I couldn't. But anyway, I'm user and teach Brazil genetics and bioethics at Londrina State University. It's kind of 500 kilometers far from Sao Paulo. If somebody wants to see me, please, welcome. Everybody is invited to come to London. And I've been working on bioethics since 2000, there, yes. 2000, and especially in genomics and ethics on research. Thank you.
Well, I'm John Weckett. I don't actually work much anymore because I've retired, but I've been working in the ethics and philosophy and science and technology for a long time at Charles University in Australia. And I've actually visited here quite often. Thank you.
Okay, it works. Hello. My name is Carla Perkins and I'm from California. And my background in my field is a bit mixed, astronomy and astrobiology, moving into bioethics. So I've been working in bioethics and teaching and crossing fields, so I'll give you a bit of a taste of where these frontiers merge. And this is my first time being in Bangkok, although I spent 10 years in Asia, in other countries sometime earlier, and about another 10 years in Australia.
Good morning, I'm Mahidol from Israel. I teach law, mostly medical law, and I've been to Bangkok a few times before, but it's my first time here in Chulalongkorn. We made it official. We're going to. And I'm very thrilled to be here. It's my first time to be in a conference dealing with bioethics, even though I'm in charge of a bioethics center in my college in Israel. And I'm looking forward to hearing whatever you are going to teach me about bioethics.
So back to me. I teach philosophy, as I said, and one of my interests is, of course, bioethics. I've been working on privacy and data protection, probably where information and bioethics kind of intersect. So welcome you one more time.
Good morning, I am Marlon Patrick Loredo. From the Philippines. I will teach at St. Paul University, Quezon City, and at AUSN. I am a bioethicist. I work more on bioethics, public policy, and global public health. Thank you.
Good morning, my name is Daniel Mishori. I'm from Tel Aviv, Israel, Tel Aviv University, where I teach at the Department of Environmental Studies, which is at the School of the Environment and Earth Sciences at the Faculty of Exact Sciences, which makes me the first philosopher ever in this faculty. And I've been doing bioethics for many years, along with business ethics and, of course, environmental ethics. Thank you.
Good morning, everyone. My name is Ananya Thaichithamongcho. I'm General Manager of U Bioethics Institute, and I'm collaborating with the Darlow and Professor Solar. Also, I'm graduate from the AUSN from PhD. Thank you. Welcome all of you to Bangkok.
Morning, my name is Thanyathorn Niyomthai from NASDA, Thailand. I work about research integrity, so I don't understand much about ethics and law on human. So I hope everyone can teach me about this. Thank you.
Good morning. It's a pleasure to be here. My name is Somsak I work at NASA Thailand Office of Research Integrity as an expert. And my background is biochemist and I just moved to working bioethics. I hope you all can suggest me in this topic. Thank you.
Good morning, everyone. It's nice to meet you. This time, first time, I'm in Chulalongkorn University. My name is Van Kim Phu. I'm from Hanoi Medical University, Vietnam. Now I'm working in medical service. Thank you.
Yeah, good morning. My name is Suradej Hongeng. Actually, I don't teach the medical ethics or bioethics, but I have to get involved in bioethics in all ways because I'm a clinician and most of the clinical parts have to deal with the clinical trial. So I'm a medical doctor and who did the job in terms of the, thanks Beryl invited me because I'm going to present the data about the gene therapy in thalassemia.
Please, Dr. Somsak.
Dr. Somsak Chunharas from the National Health Foundation. I'm a medical doctor by background and with public health. I'm already retired from the Ministry of Public Health where I work mostly in policy and planning as well as health system research. I have been involved in bioethics because I used to work with NASDA, the National Science Technology Development Agency, to manage a bioethics program for about 3 years. At the moment, I am the chair of National Committee on Ethics in Science and Technology, which is under the Ministry of Science and Technology, which again is under turmoil because the ministry is going to be merged with another new ministry, and the minister himself has just resigned yesterday because of the political complications. Thanks for inviting me. I hope to learn more. Like many medical doctors, I know very little about ethics and philosophy, but I'm here to learn. Thanks.
Yeah, thank you very much. My name is Beryl Mason. I'm the president of the American University of Sovereign Nations, and I used to live in Bangkok from 2004 until 2013 as a UNESCO regional advisor for Asia and the Pacific. Before moving, then I moved to work with Suradej in Chulalongkorn University, and he kindly has let me be a research fellow, and we work together. This is, I think, the 7th of our joint conferences between Chulalongkorn University and American University of Sovereign Nations. I want to talk soon about different approaches to human genetic manipulation. I did my first degree, bachelor's degree in biochemistry, a PhD in molecular biology and biochemistry, and then I left the laboratory the day I submitted my PhD thesis, and I've left it ever since to work on the study of people and societies and regulation. And so I'll be talking about some of this in this presentation. We have some of our colleagues on group Skype, and I'll be following through here. So I'd like to shift to the presentation. I'd like to express thanks to Ananya and Sorat for the arrangements here. And it's always a wonderful place. It feels like the home. And for those of us who walked over from a hotel this morning, we didn't notice ppm at all. But probably, we're adapted to it. Okay, thank you.
So let me introduce myself again. Basically, I'm the clinical doctor, and the whole my life right now is clinicians, and then I have worked in the lab, and I work with and I collaborate with the other countries, and I get involved in terms of the gene therapy for thalassemia in the past 5 to 6 years. And the reason why I get involved because the way I had to publish my work in the allogenic transplant, stem cell transplant, and it's been because I'm going to tell you the basic background in terms of thalassemia, and you will know what's the difference between allogenic or autologous or gene therapy manipulations. And actually, I don't teach the bioethics. But since I was a medical student and I went to the US and then I worked for the residency training and fellowship, I have to get involved in bioethics because you have to know how to write the proposals and then submit to the IRB and the ethical committees. And then especially for the human trial, you have to pass the Helsinki. That's what I learned from that. And then sometime right now, at that time when I was a fellow almost 30 years ago, I have to read and then I totally forgot. So the thing is, it depends on the clinical trial that we have to write it up, and then it's all my job, you just throw the duty for the ethical committees and IRB. But the thing that I learned from the clinical trial, the first step that you have to prove in principle that your trial or your work in clinical subject must be sound for the scientific rationale, because it's not like a voodoo medicine. So this is the feeling of the doctors. So if you say that, oh, this kind of work, it sounds for the scientific rationale, so we have to prove in principle in the in vitro and then move for the preclinical in the mouse or whatever animal models, and then go to the human clinical trial. So this is for the clinicians. So the thing is, you have to find the question that sounds for the scientific rationale and prove in principle in vitro. So I work at the Mahidol University, and this is a partner in terms of collaboration at the beginning. And for the gene therapy, I have worked with several centers in the US and one center in the Paris. So I was assigned to give the talk about the gene therapy and beta thalassemia and sickle cell disease. Since most of the slide is some kind of complicated a little bit in terms of clinician, but I'm going to go through, but I'm not going to put too much detail. So why we would like to study in terms of gene therapy in thalassemia? As you can see that the genetic disease, the most common genetic disease in the world is sickle cells and thalassemia. And then you know the genetic disease has to be carried in the gene. So the 5% of the world population are carriers of globin gene mutations. So that's why can you imagine why the thalassemia and sickle cell disease is the most common genetic disease in the world. So that's why in terms of clinicians like myself and the other are interested to cure this disease. And you know in Philippines, you may not know, they have quite a lot of patients in Philippines as well. So this is the map of the world, and you can see the colors, and the green is some kind of layers, and then when you see the colors, colorful is more prevalence of hemoglobinopathy gene mutations. So can you imagine in Thailand we have like 3,000 new births here in thalassemia major. Why? Because if you want to eradicate, I would say eradicate, to decrease the number of thalassemia patients, you have to do abortion. This is the first bioethics. So right now in Italy, I don't know who you came from Italy, you can see that. In Italy and Cyprus and Greece, because the thalassemia is a thalassa. Thalassa is a Greek name. It means like a sea. So it means that thalassemia is quite common around this area as well. And right now in Cyprus, there's no thalassemia at all because they have a very good program of abortion. So that's why there's no thalassemia in terms of abortion. But in Thailand, we are Buddhism. Even though we put a program of PND, I'm not going to talk about this too much because But just to tell you why we cannot eradicate thalassemia disease, we put the program of prenatal diagnosis, like the baby was born, and then pregnancy woman, and we check the amniocentesis, and we check the baby is diagnosed already with thalassemia. So you have to give a choice to the parents whether or not you're going to keep the pregnancies or not. Most of the time, we cannot do that because the parents want to continue the pregnancy. So that's why it's my job to treat this patient. Okay? And that's exactly the same, who came from Indonesia, and this is the belt. Indonesia, only one country, because right now I got referred from Indonesian patient, come to Thailand, we do the transplantation, we allogeneic transplant. Indonesia, the population size is a 220 million population. And then I was told that 1 million suffer with thalassemia disease, only one country, Indonesia, 1 million. So you say that this is a big burden of the genetic disease, thalassemia and sickle cell. This is not include sickle cell disease. And then you got sickle cell disease, Africa, and even in US country and Europe because of immigrants. So right now in Thailand we have 100,000 thalassemia patients that has to be treated with a hematologist. So in terms of thalassemia, the way how we correct disease, this guy is the bone, and he's my colleague, and he worked at the CEA at UC Paris, and he did some kind of experiment, try to do the gene manipulation in sickle cell disease in transgenic mice, and this is a clinical study, and then he moved to the human clinical, the first subject in the world. That was almost 10 years ago. And right now, this is the first one published in Nature. And this patient still alive, and then cured, and no transfusion, and then he's still doing okay, and no complication with the viral vector or something. So the thalassemia is a true disease, even the same mutation in terms of beta globin. This is the background, the beta globin gene. And then that the beta globin, the cell that affected is red cell. And red cell came from the hematopoietic stem cell in the bone marrow. You know the bone marrow, right? And then the stem cell in the bone marrow produce the red cell, white cell of the platelet. So this beta globin gene, even though it's expressed all over your body, in the brain, because it's a genetic disease. However, it's expressed abnormality only in the red cells. So that's why I would like to introduce the term of somatic mutation and germline mutation. You understand what I'm saying? Even though it's a germline mutation in terms of all over, but it's expressed only one somatic gene. It's hematopoietic stem cell, only red cell. But the other cell is normal. Okay, the brain or whatever is normal. But just only the red cells. And this can cause the pathology, which means the anemia, because the red cell usually usually the lifetime of the red cell is 120 days. But in terms of thalassemia, the red cell of the lifetime of the red cells is only 1 month. So that's why the marrow can, even though they're working hard to produce enough red cell, but it's not enough. So that's why these patients, they require the blood transfusion every 3 to 4 weeks for the rest of their life. You cannot cure. This is because the palliative treatment. So the refill. So that's why in Thailand, the Thai Red Cross around here, I got the data from the Thai Red Cross. For the blood donation, the red cells, 40% of the whole country of the red cell have to treat thalassemia. So this is a huge burden. So you have to get the donor to recruit the blood donor to get the red cells and transfuse the thalassemia. 30 to 40% of the red cell of the whole country you have to give to the thalassemia patient. So you see how big burden is that. Okay? And then once the patient requires the blood transfusion, iron is overload. And iron overload is damage to the whole body. So that is the cause of death. So that's why they require another medicine. Transfusion, it's a medicine as well, so-called, but they need some kind of iron chelation, and it's very expensive. So I have estimated that one baby was born with thalassemia. If you want to treat this baby until 30 years of age, in Thailand right now, you have to pay 10 million baht probably around 300 or 400,000 US dollar for until like 30 years of age, around 400 or 500,000 US dollar until 30 years of age. So this is the and then you can see that 100,000 patients in our country. So how much the money that we have to pay for the patient under the universal coverage, because right now Thailand we have the universal coverage they get for free. So this is a big burden money of the country to pay this patient for the palliative treatment. I mean the iron chelation plus blood transfusion. You see that? So this is a big burden of the country. In sickle cell disease we don't have that. Sickle cell anemia is not a big problem, even though they have anemia, but they have the problem with sickling in terms of the red cell got sick in Brazil quite a lot in Brazil. And the patient of vaso-occlusive had a problem of lung infarct. It's got occlude in the vessel, in the lungs, in the spleen, in the bone, so every single organ. And the patient usually, if they didn't get enough treatment, they're gonna die. So this is high morbidity, mortality disease. It's not like a simple disease. And then I'm gonna tell detail because the patient is anemia, and got transfusion, and then chelation. This is a picture of thalassemia. When I was medical student, around 40 years ago, we have seen this patient like this. But right now, no longer, because we give a lot of blood transfusion, and this blood transfusion has to be very special because you have to filter the white cell. It's expensive. That's what I told you, that the money that we have to pay for this patient, it's around 400,000 US dollar for 30 years of age of one single patient. It's the same thing as the sickle cell disease. This is the pathophysiology. So the rationale why we need the gene therapy, because the only curative treatment right now that's available for every single country, if they have this kind of thing, the facility, we call the allogenic hematopoietic stem cell transplant. Because the allogenic stem cell transplant, it was discovered by Dr. Don Thomas around 50 or 60 years ago, to get the hematopoietic stem cell from the other person and to treat the disease that have abnormality in the hematopoietic stem cell. When Dr. Don, he won the Nobel Prize, and the first time that he tried to do the bone marrow transplant in the beagle or mouse, that was 60 years ago. Every single scientist and doctor told him that he is so crazy. So for me, bioethics changed by time. It depends on the era that when you were born. So right now you say it's crazy, but in the next 40 or 50 years, maybe it's common things. So you must realize that bioethics is changed time by time. It depends on whatever he has said already. That's what, 1,000 years ago, it's several things that we cannot allow to do that. So it's the same thing. Dr. Don Thomas, he's the one that proved the principle that you can kill thalassemia by the bone marrow transplant by HLA. But at that time, they don't know about the HLA 60 years ago. HLA is a molecule that between the antigen between you and the others, and because at that time they don't know the HLA molecules. So that's why if you get the cells from the other and infuse to the other, and then the problem is with rejection, you won't accept. Or if you accept the cell, you're gonna cause a problem of graft versus host disease. It means the cells from the other can kill the patient with the bad reactions. So that's why once Dr. Don Thomas, he proven principle that the allogenic stem cell transplant can work, because they know about the HLA molecules, they know about the immunosuppressive. So that's why in the past 30 years, he discovered allogenic stem cell transplant 60 years ago. But in the past 30 years, we approved that allogenic stem cell transplant is standard of care for the thalassemia. Because when you learn for that, you have more and more knowledge. So that's why bioethics, it depends on the knowledge that what you get. It can change. This is for the clinician standpoint of view. It can change by the knowledge that you learn. Okay. and how you're going to make it safe and make it for all. So that's the thing. So this is, for example, my work for the allogenic transplant, and I have published the data, whatever. And you can say that the haplo and then the allogenic transplant, you must find the HLA matching. The one who can find the HLA matching is only sibling. Only sibling is mean that once the first child got thalassemia, you have to have another child without thalassemia, but can be carrier and have the HLA matching. But the chance of HLA matching is only 25%. 25%. So if you cannot find the HLA matching sibling, because most of the family thalassemia, they have only one child. They don't want to have another child because they're afraid of to get pregnant with thalassemia baby. So that's why how you're going to treat the allogenic transplant, stem cell transplant with thalassemia, but they don't have HLA matching, okay, because they don't want to have another new baby. If they have another new baby, the chance of matching is only 25%. So I'm the one because when I get back from US, I'm the one have studied about the unrelated. And unrelated has been approved in US, but at that time in Thailand not approved. So we have to send the ethical committee, blah, 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 this shortcut because in US they approved already. So we have to fly to get the donor stem cell from Taiwan. We call it unrelated. Because you have to have a pool of the donor. And then you have to check the HLA, and the chance of HLA matching is 1 to 5 to 50,000 people. So 50,000 people can find the HLA matching to you, only one. So at that time we get the HLA matching. The first time that we did the unrelated in the thalassemia almost 20 years ago. And then we success that. And then we would like to perform the father or mother to get the stem cell to the baby. But in the past 10 years, we cannot do because the haploidentical, it still have the problem, the patient can die. So in the past 5 or 6 years ago, we know how to manipulate. Haploids mean not matching. Because the baby, all of you have half of the HLA hemoglobin matching to the father only 50%. Or matching to the mother only 50% because the HLA is some kind of inherited, half from father, half from mother. So 50% matching you cannot do. But 10 years ago. But I'm the first one, and then to publish the data we call the haplo to get father or mother. And even though I get the chance of cure thalassemia, has been published already. No matter what, but sibling, unrelated, or father, mother, I get the chance 95%. However, the other country cannot do like myself because the haplo is very difficult. So I'm only one of the experts in the world that can perform the haplo in thalassemia. So that's why we have thought about the gene therapy. Because the gene therapy, you don't need matching because we need to get the own patient stem cell. We call it autologous, not allogeneic. But we have to have the gene editing to get the stem cell from the patient and correct the gene and put it back. So you don't need matching. And if you don't need matching, so you don't need that much chemotherapy because part of the allogenic stem cell transplant, you have to give the big dose of chemo. This chemo is it's a chemotherapy to kill cancer, to eradicate cancer. So it's a poison. So you have to give the big dose of chemo for the allogenic. But for autologous, you have to give the chemo, but less and less. And then there's no matching process because this is autologous cells. And then the other one, because in terms of allogenic transplant, this is some kind of alloreactivity, some kind of immunogenicity between the donor and the patient. So you need to give the immunosuppressive. So other than the chemotherapy, the patient have to receive a lot of immunosuppression. So the risk of infection, the risk of morbidity, mortality. So we have to find the good, simple method to cure the patient and have a less toxicity. So that's why if you want to deal with the new things, so the new things has to be better than the past. This is my common sense about the bioethics. If you harm to the patient worse than the previous, don't do. This is simple bioethics for myself. If you harmful to the patient than the past, don't do. So that's dangerous and that's not bioethical. Approved for me myself. So that's why we interested in the gene therapy. So and we proved in principle already that this kind of methods is safe. You don't need too much chemotherapy. You don't need at all for the immunosuppressions. But the thing that you have to concern about the long-term toxicity, that's the things. That's the things, okay? So the toxicity is acute and long-term. And if the long-term toxicity, and once you do the clinical trial, Daryl, he told you already, the liability to cover the patient who's going to pay. And how long the clinical trial going to cover. If something happened 20 or 30 years from the clinical trial, and the liability, who's going to pay? That's what he mentioned already. So that's why I will throw you this, I will give you this, the last slide to tell you for the clinician standpoint. So this is some kind of technical issue. I'm not going to go to detail.
Explain it, it's very interesting.
Oh, you want to know that?
Yeah.
Oh my goodness.
Well, briefly, yes, but you can...
Okay, so the thing is, this is the gene mapping of the beta globin gene. So the beta globin gene has a different mutation. So I'm going to explain to you how to do the gene manipulation. And then you see that this is a beta globin gene. So if you want to use one method to kill every single beta mutation, so what you're going to do? So this technique we call the gene addition. Just replace the whole gene and then for this kind of gene. So that's why this is the thing, that's what Philippe Leboche, he tried to, I'm going to show you the vector. And then how you, this is, I'm sorry, maybe, yeah, this is the gene. This is the gene. And have several, this is the gene. And then the mutation, it can be here, it can be here as well. So that's why the way how you're going, and then this is have a 200 mutation. 200 mutation. So the way how you're going to kill this kind of gene manipulation, Philippe Leboche, he just did, we call it gene addition. The way he just cloned the normal gene in the test tube. And then this kind of thing. And then we introduce the novel gene in the patient stem cell autologous. Just add it in. And then the way you want to add it in, you need to have the vector. That's what I told you. And then and then you get the stem cell, because this the hematopoietic stem cell have a abnormal globin gene. So you have to introduce the beta globin gene, and we call it addition, gene addition, to introduce into this one, the stem cell. Because the gene, you cannot clone the gene and put it in vitro and then like a simple cooking. Oh, okay, like you boil something. But you need to have some kind of vector. Because the reason why the gene therapy has been suffered for the past 30 years is not success. Because they couldn't find a good vector. The vector to carry the gene into the stem cell and set. So that's why this is the thing that has been suffered for the 30 years. Why the gene therapy thalassemia has never been success in the past 30 years. And then we need to get the vector to put the gene and then not to go to the other cells. Remember, because the gene addition, once you introduce, you understand that you got hematopoietic stem cells, and then you know these hematopoietic stem cells can differentiate to the lymphocyte, to the platelet, to the river, but we are not sure that if this vector go to the other cells, it be harmful or not. So we just need the vector to get the gene, and this gene just go to correct only the next cell. See how difficult it is? Because we want to be safe, to be sure that this kind of gene and vector just get into the cell that what we want, not go to the other. Okay? So that's why I feel he designed the vector we call the lentivirus vector. And you know lentivirus vectors differentiate from what kind of virus? HIV. It's a retrovirus, but lentivirus is the second and third generation of the retrovirus. So at the beginning we tried to do lentivirus. Everybody afraid of, oh, it can be HIV, blah blah blah. But we're not gonna take the whole HIV virus. We just get the HIV virus and then just get in terms of what we call the vector. I mean the outside, not the gene that can replicate the HIV virus. Just take the HIV, the inside gene out, like a gap, polymerase, whatever. Just the outside of the retrovirus, and they have manipulated. That's what they call the lentivirus. And this lentivirus had to get into the nucleus, not go to the other. And to go to it, someone asked me that it go to the beta globin gene. No, it can go to anywhere of the chromosome, but can express the beta globin gene. And the cells, that's what they have to get in. It's only the erythroid cells, because we want to correct the erythroid, not correct the other cells. You understand what I'm saying? And then, you know, in the past, if you know about the gene therapy, it was success 30 years ago in SCID, severe combat, I think at the UCL here. They have performed already, you know, the baby in the bubble, because this baby was born with the low function of the white cell, of the lymphocyte, and got the genetic. So that's why they correct the SCID with kind of gene therapy and the retrovirus something. However, even though they said it's safe because they take the inside of the virus vector, just only outside, but this gene, this vector, the retrovirus, once they carry the normal gene to the lymphocyte, but it causes leukemia. Because the retrovirus, if you know, the HIV virus can cause cancer, because itself is a carcinogenic virus. So that's what they're concerned, because they have done already 20 patients. I think probably half of them got leukemia later. Even though the success with the SCID, the baby survived with the back to normal lymphocyte function, but this retrovirus get in close to the oncogene of the lymphocyte and cause the leukemia. So that's why we're concerned about the long-term toxicity right now. This kind of lentivirus that I have done already and can cause the leukemia or not. That's what we're concerned. So that's why the clinical trial, they got liability to cover this patient for next 20 years if something happened. Okay? So and then I'm passed to, this is the gene. So this is the lentivirus. And this is the gene. And this is enhancer. So you just decide the lentivirus. So right now in my lab we can decide the lentivirus. So we move to the next disease. We call it Gaucher disease, is the genetic disease. So this kind of thing, once you prove it works, so we apply to the other disease in Thailand at the moment. So we just use this one and put and clone the gene, and the patient have to pay. But this one, the clinical trial, the thing is, once the patient engaged to the clinical trial, the patient do not need to pay. Okay? So that's why this is clinical trial. And it has to be multi-center. Because sometimes if it works for one center, it's not work for the other, it's not good for the clinical service. So you understand the clinical trial, right? Because in the future you're going to be the bioethics. One patient is not enough. So you need many patients to prove in principle. And one center is not enough. You have to need the multi-center to prove it works. So that's why Darren, he told you already, it has to be for all. Even though it just only safe, whatever, and then this is that what we have. And this is that what we publish in New England Journal. And we have a press conference. And right now 22 patients, 15 patients killed, no problem, and then got success, no transfusion. And 7 patients reduce transfusion. And I have heard that right now from 7, this one to 5, and move to this one to 17. So the longer period of time, more kill the patient. It looks like the transduced stem cell in some patients, it takes time to get recover and then to get success in the treatment. For the Thai patient, we have performed already 4 patients. Why I prefer this kind of method? Because I showed you already, I've done both in terms of allogeneic transplant, matched sibling, unrelated, or even haplo, the worst one, I get success 95%. However, even though the protocol 22 and 4 from Thai patient, I did myself, just only transduce, I have to transduce in US. And simple. Because the patient receive the chemo, just a small dose, and the transduced stem cells fly to me, and then we infuse with the patient. 1 month, the hemoglobin is up. You can see that. Stop transfusion 1 month, 6 months, 4 months, 1 month. Simple, no complication. It's not like an allogeneic transplant that I perform, immunosuppression, infection, blah blah blah. That's what, for me, I don't like it. So if I want to put the patient, maybe the gene therapy is the next step for the thalassemia. Okay? And right now they have a new technique for the gene therapy. I told you the gene addition. This is a gene editing. Addition, you just clone the gene. You understand what I'm saying, right? Clone the gene, and then you need to have the viral vector to put that gene into the stem cell. But the viral vector, sometimes it got stay there, and you don't know the viral vector. This is like a GMO, so the body like a GMO has some kind of things flowing around, whatever. So we concern the viral vector. Even though the first patient is safe already 11 years, there's no oncogenic whatever, but we don't know. So that's why we think that we might need to have something that can correct the gene, but no need the viral vector. So the thing is, if you want to put the gene, everything in there, you need to have the vector. So that's why this is came up another method we call the genome editing, not genome addition. And genome editing we learn from bacteria. Because bacteria, all of us, we have genetic mutation always. Every single minute we have genetic. But our body have some kind of tools to correct the genetic gene. We learn from the bacteria, like a single nucleus. That's an enzyme, and we call it nucleus, whatever, enzyme, something. And then in bacteria, the bacteria cell, this is a prokaryote. When you find abnormal the bacteria say, oh, I have a mutation. This some kind of enzyme gonna go to the mutation point and correct back to normal. So that's why this is clever idea. I think the guy who discovered this technique for the human, he's gonna get the Nobel Prize sometime soon. So he just learned from the bacteria and changed from the single nucleus to have a more efficient, more safer, and then the last technique we call the CRISPR-Cas9. It's some kind of design molecules, and this kind of molecules can get into the cells and then correct the point mutation. Okay, so that's why we call the gene editing. So right now in Thailand we have designed this technique for correct the thalassemia. This is the most common, the genetic. However, in terms of CRISPR-Cas, you need to have the vector, whatever, to get it, because the CRISPR-Cas, not to have a vector. You need to get the stem cell out of the body, and we call the ex vivo genome editing. And then we put these molecules to correct and put the stem cell back. Because we call the gene editing. Okay, and so this is the technique. And I have heard that in the future we have a new method. We call it in vivo genome editing. Dr. Beryl, this is another interesting, because the gene editing right now, there's no single disease success with the gene editing. The one that I told you, the gene addition, right? We are going on the process of gene editing for several. but you need to get the stem cell out, whatever cell you want to correct. And you decide the CRISPR-Cas9 and correct the gene and put it back. Okay, but that's hurdle because you have to get the cells out and culture and correct and put it in. And they just got the new method. You just decide the molecule of CRISPR-Cas and inject into the IV, and that molecule gonna direct to the abnormal cell. They will know because, and then they can correct the abnormal cells. So in the future, you know what? Right now they have one protocol for the I cannot sure it's a Parkinson's disease. You know Parkinson's disease, and then they have heard that 20% of Parkinson's disease abnormal gene, we call it like a GB2 gene in the brain. So right now they decide the gene editing like this, and then they inject into the body and get into the brain and correct. So they hope that Parkinson's disease in the future you can treat with the in vivo genome editing. So the next you have to live longer. Maybe you can get treated with this kind of thing. We call it in vivo genome. This is a new technique. We call it in vivo genome editing. And then the knowledge of the gene editing. I think in the future you have to learn about the stem cell. You remember the stem cells because the knowledge about how to live longer with the good quality of life and disease. The gene editing and the stem cells has to be merged. And I believe artificial intelligence, it's another knowledge, has to be merged. So the way how we live longer, the aging process, that's what I predict in the next 10 or 20 years from now, because the knowledge machine or the learning machine right now is so fast. So I think the health science in the future, the knowledge about the gene editing, the knowledge about the stem cells, and the knowledge about artificial intelligence will be merged together. So that's why the bioethical people like you have to learn about this and then get used to this kind of things. And then the stem cell, and let's just give you a little bit of the stem cell. So all of the body, you have the stem cell in your body, but it's specific stem cell to the organ, like a neural stem cell, liver stem cell, and kidney stem cell. And this stem cell we call the somatic stem cell. But another stem cell we call the embryonic stem cell or germline stem cell. You just get by this one, embryo, and this one has been bioethic in the past 20 years. You remember the past 20 years? They say, oh, you cannot do the embryonic stem cell, blah 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 blah. And right now, every single country you can allow to have a research in embryonic stem cell. Because 20 years ago, banned. See, I told you bioethics, it depends on the time that you work. It's dynamic, it's not stable. So, and then they allow you to, the way how you develop embryonic stem cell is mean that the embryo around 5 to 7 days, you just get this inner cell mass out and then expand. That can be embryonic stem cell. This is the embryo. You know, the IVF, right now the technique, 30 years ago, before 30 years, the IVF banned. IVF in vitro fertilization because you need to have the natural baby, not some kind of in vitro something. And this was banned 30 years ago. And right now, boom, it's fashion. And then the one who has the problem of infertility, you have to use but this is a blastocyst 5, and then you just get inner cells. And then you culture in terms of fibroblast, and you will get the embryonic body. And this embryonic body can differentiate to any single organ of your body because it's embryonic stem cell. Because the somatic stem cell, even though it's somatic, but it has the potential just only for the organ, that's what belongs to. Because all of us has a somatic stem cell, the neural stem cell, the heart stem cell, but this one is embryonic, it can differentiate to any kind of cell. But so far, I have not heard that the embryonic stem cell can be treated in clinical use. Why? Because embryonic stem cell, if you give to in animal model, some animal models get the wrong differentiation. because you might have some kind of liver in the brain and the kidney in the whatever. It can be wrong differentiation. But in the future, I think it may be working because the AI, the machine learning, it will be so fast. You can control the genetic expression, and you can tell this kind of cells what can differentiate to whatever cell that you want to be, what want it to be. The next one, because the embryonic stem cell, they concern too much. You remember, because you have to get the embryo and then take it out. And then some of the things they say cloning. Have you heard of cloning? You know cloning what it is? Cloning is just, you just get the egg like this and then take the nucleus out. And then you get the chromosome from the skin or whatever, the DNA, and then put it in. And then you get cloning. So that's the way how to get cloning. But so far, right now, the human cloning not success yet, because that's only animal, okay? So Yamanaka is the first one that can develop like an embryonic stem cell, but it's cloning from you. They call IPS. It is induced pluripotent stem cell. They just get the skin, and then they put the 4 factors, because they learn already this gene, the transcription factor. Once you put it in the skin cell, it can change to be your embryo, the same thing like you. So that's why they call the embryonic stem cell. So that right now you can have your own embryonic stem cell and then storage in the bank. In the future, if you want to get the new liver or new cells, maybe you can do that. That's another thing you have to think about for bioethics in terms of stem cell research or regenerative medicines. Because this is, you can get the IPS for your cells and you can differentiate here. Because right now I differentiate, I can do, this is for example, my work in the lab. We can differentiate from the skin to the red cells, to the white cell. We can differentiate skin to the kidney. I can do in my lab. We can differentiate skin for the brain. We can differentiate to the heart. I can see I didn't show you. We can change the skin to the cardiac myocyte and have a movement like this in the lab. So in the future, the stem cell may be another things. So that's why you remember I told you already, the knowledge to improve the quality of life in the future is the gene editing, is the stem cell regenerative. You have to be prepared for the bioethical people. You have to running so fast to keep up knowledge. So that's why you can prove or not prove of them. And bioethical concern for me myself. The first step, if you want to do anything, your knowledge, that one you want to prove, it has to be scientific, rational, sound. If it's not sound, don't work. Because right now they have a lot of voodoo about the stem cell research, you know what I mean? And they get the stem cell and help. I didn't say that it can be done, I didn't say it can be done in the future, but for now, it not be safe. Because the knowledge is not enough. But in the future, maybe. And then once you prove already, you have to prove in vitro, in the test tube. And then move to the preclinical, animal models. An animal model is a bioethic as well, right? Because they have an animal people that concern the bioethics about the animal. And then in the clinical trial is a must. Because right now, you can go to anywhere, because right now they have a pressure from the country like the EU, US, or China, or Japan. They develop, but they didn't pass the clinical trial ethics. So they want to go to Indonesia, Bangladesh, Thailand, there's no knowledge about bioethics. And then they have, because the government, they pressure so much, because the research has to be some kind of use, to be used for the clinical use for the future. But in terms of bioethical people, they say, oh, it's not the time yet. So that's why this researcher from the Japan, for the China, they done already in the mouse model, but clinical trial, it takes time. And this kind is a business, oriented. So that's why they're gonna come and talk to the country that there's no regulations, no bioethical concerns. And then they want you to get approved for the clinical trial. Or even though, and some of the doctor in some of the country, not clinical trial, but the clinical use. So that's why you have to teach your people, I think, these kind of things. you have to not just only from the country that's developing country, the one that size is getting more knowledge. The science is kind of better in some country, but in some bioethical is not concerned. That's what I think. This is some kind of critical issue. And then the somatic gene manipulation. The one that I did is somatic gene to get the stem cells and then the manipulation. But however, germline so-called manipulation is not allowed at the prison. Have you heard the news in China? They get the blastocyst, they do the CRISPR-Cas, they collect at the embryo, at the round egg cells, and they claim that that embryo is resistant to the HIV. And then that kind of scientist, he's disappeared because he claimed that he can CRISPR-Cas the blastocyst. That's the germline, not the somatic gene manipulation. You might not know, in 20 years later, it can be allowed. Because we want the baby that's healthy. That's what he told you already. So the bioethics is changed from time to time, from knowledge to knowledge. That's what you can learn already. So that's about it for the scientific point of the definition about the bioethics in this kind of gene manipulation.
Thank you very much. It may be still good to have your questions up on your bioethics question. Please. You made some comments about bioethics, so now we have to ask you a few questions. I think I'd like to open for discussion, but first, I think here the bioethics may be the same, but the legal regulations may change. Okay, so if the bioethics is the love of life or curing of sickness or trying not to do too much harm, that's the same. But it's like the etiquette, the legal regulations and what's customary accepted. This may be changing, the trendy things, as you mentioned.
Thank you very much.
So, so the bioethics, of course bioethics may be evolving, and I've written something on the evolution, and I actually now I think it's a circular evolution, it's not linear. Circular evolution of ethics, and then Somsak's going to talk about ethics in Thailand as well. It'd be very interesting too. But do you have any questions? And also, I think I'd like to just make also a reference for you, since our colleague from the National Commission of Ethics in Indonesia, Agus, is here, Vice Chairman. That's actually a commission being on for several decades. So actually, Indonesia was earlier than Thailand in terms of ethics regulation, but it's quite a large country, and maybe Agus would like to make some comments on that situation as well. Agus, would you like to make some comments, and then we go into general questions?
Yeah, it is an interesting session that I follow your session in Dr. Suradej. Yeah, because it is the future of our destiny, because we have to what we call it the nowadays issues human enhancements, something like that, or another issues like eugenics issues that people tend to improve the self, the body especially, and also the brain. We have also issue of brain stimulation, deep brain stimulation, something like that. So the genetic editing is like the
we correct only somatic cell at the moment.
Yeah, that's this is in line with Indonesian, and I think it is in line with other international norms that only somatic manipulations already allowed, but the germ and the sex of the gene or the sex it is not allowed up till now. But yeah, this is the lack of paradox, because due to the technology, it is we call it the feasibility aspect. But in the bioethics, we should consider the necessity. So the possession means that it is our balancing the necessity aspect with the feasibility aspect. So usually that if we learn from international institution, we can learn faster if we learn from FEO and compare with the WHO. Because if we should implement it in the human, it is many of considerations. So this is my comment that I'm agree that this is the challenging for balancing the advancement of technology with the humanism. Thank you.
Thank you. So thanks. I like your speech at the moment. Bioethics never change, but the regulation has to be changed from time to time. Thank you very much, because the bioethics that what Daryl said is has to be safe. He has a definition already. And definition is perfect, but the regulation, it has to be follow the bioethics, and the regulation, why? It's time to time. It depends on the scientific rationale. It depends on the knowledge. It depends on whatever learning machine at that time it works or not, or it's safe or not. So that's why I like the thing that you work. So that you correct my thought is that bioethics never change, but the regulation is changed from time to time. It depends on whatever, and even the regulation, it depends on country to country. That's the thing. It depends on nation to nations, and regulation, it depends on belief to belief. That's what I don't know, this is the way how I learn. I'm not a philosopher, but this is a common sense. Bioethics never change. The regulation, it depends on your belief, it depends on the religious, it depends on the knowledge that what you have learned in your country, it depends on the tools whatsoever. I don't know this is correct or not. That Daryl can, maybe you can correct my thoughts.
Yes, no, well, I think that everything is evolving over time, and so we do actually see the evolution of ethics and law and science. So the fundamental principles may be these, is what I'm saying, they don't necessarily change, yeah, the principles of love and law and there's history, but it's like the definition of death. Where do we look for death? Do we look at the heart stop, or the liver stops, or it's turning green, or now we look at brain death, or in the future we might look at virtual death and all sorts of things. So the essence is what is acceptable is changing, but that's not necessarily the bioethics, but it's thank you for accepting that. We have a few.
Yes.
Okay. Okay, behind me first. Please go ahead. Yes. And please use the mic. It's good to have a student talking.
I'm just concerned since each country can have or can implement their own regulations about gene editing and gene manipulation. Do you think the UN needs to create a committee that looks for the regulations or to standardize the regulations worldwide?
Well, it's not regulated, but it has to have some kind of core regulation or something. Yes, of course, you must have some kind of WHO or UN something for the clinical trial conduct. But you cannot tell the single detail, because this one I told you already that it depends on country to country, belief to belief, and then the knowledge from each country. Let's say the gene editing, if it's happened where it's somewhere in the forest and then nothing happened, you cannot have the background of that, maybe it's not regulated. It's not allowed to do because the facility that you're not enough, you know what I mean? Because this kind of thing, you need to have some kind of standardized of clinical trial, the CPG, the laboratory, whatever. So its regulation is different from bioethics. That's what I learned from him right now. So you say that, well, the regulation, yes. like because if I'm the clinician right now, I have to pass the GCP, and my hospital has to be accredited by some kind of certain level, because you cannot perform this kind of thing in the village hospital. Totally dangerous. You know what I mean? Because you need to have some kind of, that a lot of facility that can afford, that can support for the clinical trial even. And right now, I'm gonna do the clinical use in Thailand, this kind of gene therapy. But it's just only some certain hospital. I'm not gonna allow I'm not gonna open the clinical use for everywhere in Thailand or something. It has to be regulated.
Okay, Daniel, please.
Thank you very much for your lecture, and I would be happy if we could get the presentation also again.
We can have that.
And my question is like this, because you said that bioethics is the same, but regulation changes, and I wanted to ask about science, because presently we have a situation in which everything that is done according to certain formal structure is being referred to as science. So if I'm doing something in a laboratory, I have any kind of hypothesis, and I use modern technology in order to test this hypothesis, whether or not this hypothesis should have been tested in the first place. So the question is, first of all, is the science the same everywhere? I'm not sure. And secondly, what should come first? What should come first? Bioethics or so-called objective science?
Interesting question. It's like a chicken and egg. It's like a chicken and egg. You cannot say that this come to first or whatsoever. You know what? If you can think about the past, you can go back to the 2,000 or 1,000 years Aristotle whatsoever. I think it's the same. The ethics and the science, you cannot say that this is first, this is second. It have to be at the same time, I believe. And in the science, yes, everywhere is the same. But the knowledge the ground knowledge of each country, of each people is different. So that's why, science is fact. But the thing is that you have to learn how to get that fact. Otherwise, Nobel Prize is only one people a year. You know what I mean? So that's the science. Science is the same, but the way how you discover, it's different. I mean, the chance of discover or whatever, because science is fact. For me, myself is fact. But sometimes you don't know the fact yet. It has to be proven by some kind of whatever. Yeah. But this is an interesting question.
Sora, did you have a question? Anybody else have Angelica, please.
Professor Suradej, very impressive presentation. Thank you. I'm Dr. Bailon. I'm from the Maritime Academy of Asia and the Pacific. And in addition to being a bioethicist, I'm also a biochemist turned into a maritime director. Now, this reminded me of, cancer-causing things. Because when you mentioned about gene manipulation, this reminded me of the what if during the manipulation of gene there will be a mispairing, the AT, the GC, GC, if I remember it right, adenosine, thymine, and the cytosine, guanine. So when you did the gene manipulation 20 years ago, the patient that caused leukemia, the patient. So it means in the process of scientific studies, there might be a change in the pairing of this AT and GC. Now, so my concern here is how sure are you as a scientist that in the process of doing such clinical or experimental studies?
I know what you.
Yeah.
I know what is your question.
Yeah.
Yeah. Thank you. This is a very good question. Even though it's simple, but it's a very good question. Because the thing is that we use the gene manipulation. We just add the gene, and you're concerned that this kind of gene that can get some kind of mutation in the future. Well, of course, all of us, the gene inside our body can mutate every time.
Yes.
So otherwise, we're gonna be forever. You know that cancer, it's the disease of aging, and it's the disease of mutation, because it's the disease of the genomic instability. You know biochemist already. The gene that will be put. Oh, of course, in the next future, it can be abnormal. No problem with that. But the thing that we concern is, right now, the one that I use is a vector. That's what I concern vector. The vector is a retrovirus. I mean, the derivative. We concern that this vector is a virus. You know, virus can cause cancer. And this virus, if it get into some kind of oncogene and sit there for longer period of time, it can cause cancer. Of course it can happen, but the thing is, I think the genomic mutation or genomic instability that what you have put in is going to be very low. But the thing that concerns is vector. Okay, you understand what I'm saying? So that's why we want to do the gene editing. Because the gene editing, we don't need the vector. We just have some kind of tools and get into and eat it. You know what I mean? But right now it's not success, because the gene editing, if you put it in the body or in the cells, you want to correct this point. But we concern that we call the off-target, because this gene editing can go the other and can correct and can cause cancer. You know what I mean? Yes. So we concern right now acute toxicity, but the long term, I'm not sure. For me, it can happen, the genetic instability. But you have to wait, because right now we solve the problem right now. But in the future, 30 years from now, of course it can be, but I cannot guarantee. Yeah.
But based on what you presented, I think it's being successful now.
Yeah.
Because initially there was there was leukemia, but now leukemia is gone now.
No. This is thalassemia. This is the one that I treat is thalassemia.
Thalassemia.
Yeah, but the one that I told you that the retrovirus for the other disease can cause cancer, the leukemia. Okay. You understand what I'm saying? Yeah. That's what we concern.
Very interesting. Thank you.
Well, thank you. Okay, what last question, Tamia, please?
Sorry, I'd like to talk a little bit. Thank you very much for your presentation.
Please use the mic.
An eye-opener. Just one small question. Who's going to bear the cost of your mistakes?
Cost of the treatment?
Cost of mistakes.
Of the science mistakes.
Science mistakes? You mean liability?
Liability to somebody. I don't know the rule. But,
yeah, of course. Yeah. That's what I mentioned already. Who's going to pay the cost of mistakes? So the thing is, the clinical trial, usually we have the liability to cover, because the one who sponsor, you have to buy the liability from the insurance to cover for how many certain level. So that's why you have to take a look when some kind of ethical committee, because ethical committee have to concern this one, the liability, who's going to pay how much money. Yes, that's to cover by the insurance. So that's why the clinical trials is very, very, very expensive, because they have to buy the liability from the insurance. Very expensive. So that's why the clinical expensive. It come up for the clinical service very expensive. So if this one is available in Thailand, who's gonna pay? Because it's expensive. The clinical trial is expensive. So it's like a chain. You know, expensive of science, technology, expensive of, mouse model, expensive of clinical trial, expensive of liability, and one clinical use, who's gonna pay? Who's gonna pay?
Shouldn't it be the pharmaceutical companies that cover that kind of cost?
You mean the clinical trial? Yes, but once clinical use, no.
And specimen?
It depends on patient. It's up to the patient. You have to accept that.
Thank you very much. We're getting into a great discussion, which we will continue later, I think. Yeah, thank you.
And CRISPR-Cas9 work. And you can see also that really we are not talking, I think this is a little bit helpful to the discussion in any way. Probably you've discussed this as well, that when we talk about gene editing, it's not only about the editing of humans. That's kind of a late comer in a certain sense. We're talking about all kinds of living cells, all kinds of living organisms that can be edited in order to delete or alter how genes function. So on the next slide, slide 5, just some of my very basic layman's understanding of CRISPR-Cas9 science. And I think the last presenter seems to be the real scientist in the room, so apologies when I make these mistakes. But we're talking about RNA-guided engineered nucleases that bring simplicity, efficiency, and versatility. It's really only since 2005 that we have this kind of, let's say, biological tool in order to begin to experiment with gene editing and understand what its possibilities are, as well as perhaps also understand where the concerns or threats may be. So this gene editing tool gives us a capacity to modify genomes rapidly with precision and in a predictable manner, and that's important. It's also simple. It's a rather easy tool to be used, I believe, by biologists, and it also allows us to repair non-functioning genes and create new variants. So you can see just from this, it's a really enticing and hopeful picture. And it achieves a high efficiency genome engineering using a biological tool that's simple, inexpensive, and reliable. And this particular method for, let's say, getting involved with the human genome has been going on since 2005, and it's become increasingly, let's say, since 2011, 2012, really much better understood. And we've already seen that there are a lot of studies being carried out about bacteria, plants, and animals, and even human gene lines. So what happened in Shenzhen with Dr. He, we could say was perhaps not entirely unexpected. In fact, this entire international moratorium and the outcry that we've seen almost suggests to me that it was in some ways expected. In other words, we knew we had the possibilities, we knew we had the tools, and we knew that this kind of experimentation was possible. And we live, perhaps not in a huge world, but a rather large world. So in slide number 6, I talk about what are foreseen as advantages of gene editing. And again, you probably discussed this. Gene editing gives us a way to model diseases using cell lines and animal models, and to see how then we can interfere or intervene in the biology of those diseases. It gives us a way to improve the production of food and reduce plant diseases. And this is important because it really has to do not only with humans and not only with animals, but also it can have to do with vectors in disease, and it can have to do with food production, which will be perhaps something of importance for us. It's a way to address human and animal health, including genetic diseases, non-communicable diseases, so as cancer, as our last speaker discussed, and communicable diseases like viruses and bacterial infections. So it has a wide range of possibilities with regard to human health, and with regard to animal health, and with regard to the provision of food. Now, in the European Union, the French, in 2018, went to the European Court of Justice and challenged the use of mutagenesis and whether or not mutagenesis was to be conceived as a genetically modified organism. And quite surprisingly to many, many people, the European Court of Justice came down and said that the use of mutagenesis, which I believe is something different than gene editing, does come under GMO, the GMO directive, which is a European law. Insofar as the techniques and methods of mutagenesis alter the genetic material of an organism in a way that does not occur naturally. What's important here, too, is that the court said that because this particular technology was not in use prior to the law coming into effect, or the law being voted on in 2001, that this had to be considered as a new technique that did require it to go through the procedures for genetically modified organisms. The genetically modified organisms are not forbidden in the EU, but those classified as genetically modified organisms need to be especially looked at by the regulators before they're allowed to be marketed or put into the food chain or into other areas of life. Now this decision by the European Court of Justice really shows a division in society. About 180 German scientists wrote this article in the Süddeutsche Zeitung in August of 2018, stating that German regulators shouldn't trust test biotech and other anti-biotech lobbyists to guide the public about CRISPR gene editing. And above that, I just put a picture of a protest in Germany against GMOs and against this kind of, let's say, interference in cell lines or in the genetic nature of organisms. In the next slide, slide 9, I just show you simply what the GMO directive, the genetically modified organisms directive from 2001, how it defines genetically modified organism. It says a GMO means an organism, and here's very interesting, with the exception of human beings, in which the genetic material has been altered in a way that does not occur naturally by mating and or natural recombination. So it has to do, and this is pretty much the way the court went as well with regard to mutagenesis, that it has to do with alternating the genetic material in such a way that it is not done naturally. But here, interestingly, the GMO directive specifically left out human beings, and that means this was going to be legislated somewhere else. And this is legislated in Europe now in the European Clinical Trials Regulation from 2014, which has yet to be implemented, which thus means it's not a law yet. But nonetheless, we can take it as law here. And here in this EU Clinical Trials Regulation, it says no gene therapy clinical trials may be carried out which result in modifications in the subject's germline genetic identity. And what you see here are two things. Firstly, of course, it's clear, okay? Gene editing or clinical trials using gene editing would not be allowed under the legislation, the current or coming into effect whenever we can get Brexit sorted out, let's say, European regulation here. But also this idea of genetic identity, I believe this kind of refers, in a certain sense, to the Convention on Human Rights and Bioethics, indirectly, which comes from the Council of Europe, which is kind of outside the EU. So it's a very strange thing here. But I think this idea of identity, this is not really such a strong scientific word, I believe. It's more of an ethical word, or a societal word, or how people define themselves. Now, I'm just going to say something here on this slide, number 10, the title, Strong European Union Position. Whenever I go to Brussels, and that will be in about an hour from now, I suppose, and I go into a European institution, be it the European Economic and Social Committee, or be it the Committee of the Regions, or the European Parliament, or the Council of Europe, or the European Commission, they will tell you every time you walk in the door, leave your ethics in the coat. Okay, don't bring your ethics in this building, because we will not have anything to do with ethics. Ethics, according to what people say in the European Union, belongs at the member state level. It is not a European competence. It's not foreseen in the Treaty of European Union. So ethics is something for the member states. So it's very rare that you will see in European legislation any remark toward ethics, because that's when the member states will get very upset and say, no, that's our prerogative, that's our priority here. So I'll show you how, but with certain kinds of scientific research, that means also, and in this case with clinical trials on gene therapy, the European Union will be very careful about allowing or not allowing certain kinds of research at the European level, or funding it at the European level. So if you look at the next slide, slide 11, where I say looking back here, we're looking at the Convention for the Protection of Human Rights and Dignity of the Human Being, the Convention on Human Rights and Biomedicine, or what's also known as the Oviedo Convention from 1997. Now this Oviedo Convention comes from the Council of Europe. The Council of Europe is not part of the European Union. It's outside. It's made up of like 45 countries, so it's quite a different body. But it was a kind of predecessor, and it's in Strasbourg, not far from the European Parliament that's in Strasbourg. So we have some complications here. But the Oviedo Convention does hold a lot of sway in Europe with regard to bioethics. And this is what it says in 1997. It says, an intervention seeking to modify the human genome may only be undertaken for preventive, diagnostic, or therapeutic purposes, and only if its aim is not to introduce any modification of the genome of any descendants. So now we're really getting into murky waters when we're talking about genetic mutations, or when we're talking about gene editing. So this is from 1997. This is prior to a lot of the technology that we are going, because the technology goes very fast today here. So this makes it a little bit complicated for us here. Also, just to say something about this, in 2018, the Bioethics Committee of the Council of Europe said about this particular sentence here, that this was not really, let's say, a required statement, but more like a guidance statement for discussion. But you know, the Oviedo Convention is a convention that people have, that states, governments have signed up to with their signature after ratifying it in their parliaments. So it's a very strong document in the background of the discussions we have. So with regard to European Union funding, the Horizon 2020 program is the title of the program for the general scientific funding in Europe. There are some restrictions. So the European Union, at the union level, this does not mean at the member state level, but at the union level, the European Union will not fund research activities aimed at human cloning for reproductive purposes. The European Union, and this is usually the Directorate General for Research and Innovation at the European Commission, will not fund research activities intended to modify the genetic heritage of human beings, which could make such changes heritable. So that goes clearly right back to the Oviedo Convention. But interestingly, they insert an exception. That is because if you know European politics, you know that the European Organization for Research and Treatment of Cancer is rather powerful here. So except for cancer treatment of the gonads. So they make some exceptions already. And research activities intended to create human embryos will also not be funded at the European level, solely for the purposes of research or for the purpose of stem cell procurement, including by means of somatic cell nuclear transfer. However, in some member states, this is allowed. For example, in the UK, and it's going to be a member for another 58 days, I believe. So, in other words, we have a complex situation because in many member states, especially those who signed up to the Oviedo Convention, they would not allow research on human embryos. And some member states, because of their history, are also very concerned about this kind of involvement in, let's say, genetic modification, which can often be seen as eugenics, which of course has a very negative connotation, especially in Europe. In the next slide, slide 13, the European Commission will additionally not fund, although it's not legislated, it will not fund human germline genome editing research. So what we're talking about to a certain extent. And research leading to the destruction of embryos. Though again, in the UK, they will fund this. And the European Commission, however, does fund several projects that relate to gene editing. And those are in the fields of cancer, in the fields of advanced therapies, immunodeficiencies, rare diseases, and in other fields as well. And I could even indicate if I wanted to specific projects that are currently funded. So slide 14, I want to just look forward here. I have 22 slides in total, just so you know when you need to yawn. Looking forward, I take this quotation here from a very interesting report from the Federation of European Academies of Medicine on the application of genome editing in humans. And I recommend this to you if you're interested, because it gives a very good overview, really, of Europe. And the European Academies of Medicine is located in Brussels. It's it's located in the building of the Royal Academy of Medicine, right across from the palace here, the King's Palace here in Brussels. So it's quite a nice place. And they, this actually comes from a meeting at the French National Academy in 2017, but it's part of the European Federation. And it says, from an ethical perspective, European society is generally supportive of the future potential of somatic cell-based therapy. So, now you're talking about scientists here and medical doctors here. And they are going to, if you read this, you will see that they really are interested in what we are going to do with gene editing in the future. Although, it says, it does appear that some countries may be more advanced than others in the scientific application of such genome editing. And then they conclude that there is a real opportunity for the European biomedical community to take the lead in this field for the benefit of Europe's patients and those from other countries. So you can see this is really going strongly down the line of we need to be involved in the genome editing of humans. Of course, this is not an official, let's say, legislated government position, but this is from the Federation of European Academies of Medicines. So, how do I understand the ethical perspective? Well, for me, ethics is, in the first place, a descriptive activity. And I hope, in the last place, it is also a descriptive activity. And I think in ethics, there's little room for shoulds and oughts and musts, because they generally won't work anyway. They can be helpful. You can say should or ought, and you can even say, in certain circumstances, must or must not. But these are more, let's say, guiding principles than actually principles that really show us how to behave as human beings. So the editing of human, animal, plant, and bacteria genomes is scientifically possible and being carried out at an accelerated rate. That's simply a fact. We should focus on the possibilities the scientific methods provide toward the future. And those possibilities, and now we have to be careful, include promises, and great promises for health. I mean, there are many, many Europeans and many, many more people in the world who would like to have their health condition addressed in a way, in a much better way than we're able to address it today. This includes people who have cancer, people who have HIV, TB, people who have malaria, people who have rare genetic diseases, all kinds of areas in health that gene editing holds promise for, and patients would clamor at the door for. So there are promises, and these are real promises, and we have to take them seriously. At the same time, these possibilities also include threats. There are risks, and the risks are real. They're not fake, and I heard you talking about them just now. For example, missing your target. So we really have to have a very good understanding and a very good description of what's going on. And that understanding and description can not only be among scientists. That cannot work in our societies today. And it can not only be among scientists and policymakers either. It really has to be an understanding that we take on as a society. And that means we have to engage our societies, and not just ethicists and scientists, let's say, in this discussion. Individual scientific gene editing projects should be carried out openly and examined for their safety and efficacy, as well as for their impact on individual society and the natural environment. So it's very important that we create open science. I think this is the real ethical imperative here, that we have open science. And open science will only be achieved when we create the space for it, and we create the comfort room for it, and where scientists trust others to be involved in decision-making about science. Slide 16. So what are the ethical questions? The editing of human, animal, plant, and bacteria genomes is scientifically possible. So now I'm repeating myself, I apologize. So what is required? Slide 17. What is required for a societal conversation? I think we need open science. And I think that's the most important thing that we can have. We have open science. What does this mean? This means we need open publication. And this is a crucial matter, a very important matter today. Plan S is very important. Open pharma is very important. We need to find a way where scientists put their information out. Also, I will say here too, we have new ways of reading and writing. The universities which were founded in Europe were founded on the basis of reading and writing. The liberal arts, which came from the monastic schools and from the cathedral schools, the trivium, the most important three subjects of the liberal arts that founded the university was grammar, grammar, dialectics, and rhetoric. Those three are the three basic liberal arts, and they're all about reading. How do you read? So today we read differently because we use digital media, and that's a huge difference. And it also opens up to a much wider audience than before, so that's important. So we have to make sure that people can read science. That is very important, and that means we need open publications. How we pay for them, I don't know, but it cannot be pay-per-view. It cannot be that we run our universities. The budgets of our universities go into putting volumes and volumes of journals on their shelves at huge expense, or online at huge expense. That has to stop. So Europe is very active in that, and you can see that with regard to Plan S and the Coalition S as well. And also pharma, there are 6 pharmaceutical companies now involved in that, two of which now have agreed to that all their science will be published as open science. So we are going in a good way. And there's a discussion in the US and the NIH has a policy on opening up science. We need responsible researchers. We need researchers that are scientifically knowledgeable and skilled, qualified if you want, that are responsible and have a sense of responsibility, and that demonstrate integrity. We need open dialogue, the kind of dialogue you're holding today in Bangkok, and I wish I could be a part of. We need competent ethical review of research proposals. We need good ethics committees. So we didn't start FERCAP or SIDCR or all these other FERCIs and all these other organizations we set up for no reason. We did this because we need really good ethical review, because the scientists need it, and they want it, and they will rely on it when it's good. We need community and patient discussions, and we need sound informed consent procedures and information. And we saw this in the discussion in Hong Kong and everything around what Dr. Hay produced. And the next slide, 18. So what should we expect from a conversation? We should expect a sharing of values and perspectives. It doesn't mean we will attain, and we shouldn't even expect or think to attain the same values and perspectives, but we should share them so we understand each other. We should share our expectations, we should share our concerns, and we should share our worries. And we should exchange the information we have on diseases and on science. We should engage to listen to and appreciate varying perspectives, and I should say varying values. And we should work towards a growing mutual understanding alongside an increased appreciation for differences. There are 2 values in the world, as far as I'm concerned. The second one is health. The first one is peace. Peace is the most important thing. Health is the best way to ensure peace. So those are the things that we have to consider whenever we enter a discussion. 19, slide 19. What outcomes we might expect from an international conversation on the science and ethics of gene editing? We should expect, and this ethics will participate in this first bullet point as well, improved and more precise scientific methodologies. Both, we should have better science-oriented, better toward patients' need, increased personalized medicine, and of course, anything to do with the genome and getting us with a better understanding and a better intervention in genetic medicine will will lead towards personalized medicine and more attention to neglected and rare diseases. And I think this is really holding great promises there, if we can get there with gene editing. A better understanding among scientists of community, societal, and environmental interests and concerns. It's it's important that we educate the scientists as well outside of their domain, just as we need to be educated inside of their domain. And we need greater access to science and health technology. And that means we need medicines. When a medicine has been shown to be safe and efficacious, when a medicine is, let's say, on the market somewhere, patients need access to it. And that means patients everywhere. So we have to, and we're going to do this, we're going to reduce the cost of science, the cost of medicines and the greater access to medicines through open science, through open publication, and through advancing health technologies. Slide 20. What outcomes we should not expect from an international conversation on the science and ethics of human genetic? We shouldn't expect, I believe, and I could be entirely wrong, and it's not because I'm promoting it or not promoting it, it's just that I think this is the reality of the situation. We should not expect that gene editing will slow down or stop in experiments on humans, and including experiments on embryos and fetuses. We should not expect it. We should understand gene editing will continue in animals, humans, plants, and bacteria. We should also not expect that ethics will get ahead of science. In other words, we won't be able to say today what science can or cannot do tomorrow. I don't think that's possible because science will always go first, and ethics is about how you behave. And until you know what the behavior is, you cannot describe the ethics of it, I believe. Also, we should understand that we will reach a consensus on what is permissible. Oh, sorry, this is a wrong sentence. We will not reach a consensus on what is permissible and what is not permissible. The word not should be there. I apologize. We will not reach a consensus. And I think it's impossible to reach that consensus. I also think we can say Belgium will decide what it does, and Germany will decide what it does, and the UK will decide what it does, and Uganda will decide what it does, and China, and Thailand, and the Philippines, and Japan will all decide what they do. But that's not how science works. We share science now on a global level. So we're going to have to share the ethics. And if we say we want only a consensus in Belgium on gene editing, it will be ethically empty, because ethics involves all human beings. Slide 21. What we can achieve from an international conversation on the science and ethics of gene editing. We can achieve improved science, and we will. By including ethics, we will improve the science. And I mean we will improve the science, as the methodology, as the technology. We will improve the ethics. And we need to improve health literacy, which is what I will discuss in a few hours. We really need to do that. And health literacy, I insist, is not just about the guy on the street like me, or my neighbor, or the person down the street who's a patient, or whatever it is. It's not about poor people, and it's not about rich people. It's not about those who are laypersons and those who are leading scientists. Health literacy is for everyone. And all of us have to participate in that. And we need to share what we understand about health. What I understand about my health, even though I'm a complete layperson, is of enormous value to my doctor, and it's of enormous value to the researcher who says to me, do you want to participate in a clinical trial? We need, and this is important here, we need a sober and good understanding and appreciation of, as well, risk. So what is risk? In Europe, this is a very important concept because it's part of the GMO directive. So we really need to understand what risk is and how it's important. We need improved health for humans, animals, and plants. We need an improved natural environment. And we can expect as well, only reasonably, that there will always be a trade-off between what we consider as a pristine natural environment and what our needs are as human beings for health, and that means for food and for medicines. And importantly, if we have a good conversation, we will really learn to respect one another in our agreements and in our disagreements. So just to conclude, this is what I believe. This is why most of my life has been devoted to research or to ethics committees. Because I believe ethics is a gateway to science and society. If you don't have a good ethics committee, you won't have research. If you don't have research, you won't have good health. So you need good ethics to have good research, to have good health. And health has always been my objective, because health is the second value, and health is what gets us closest to peace in the world. Second clue, well, when you understand that ethics is the gateway, we also understand that science propels ethics in its understanding of persons and societies. We understand ourselves better, and we understand our world better because of science. Science improves ethics. An open and structured dialogue between ethics and science on gene editing is required for an ongoing appreciation of ourselves and the world. And I thank you for listening to me.
Thank you very much, Francis. Does somebody have a question or two? Yes, please.
Thank you so much for that very inspiring and very to the point discussion. The one thing that comes to my mind in terms of health literacy in its globalization and collective society is the politicalization of health literacy and how much economics and politics plays into the ability to have health literacy. Just for an example, working in Hawaii, we have the indigenous population, and because of economics, their health literacy, they have a very powerful indigenous knowledge, but the economics of every other corner having a fast food place on the corner and lack of access to good groceries ended up with 300-pound and 500-pound Samoan people. And so that interplay between politics and economics and health literacy seems to be a really imperative one for us to consider.
Thank you for, I don't think it's a question, I think it's a teaching for me, so I really appreciate that. I tell you what I think, but it's only what I think, and it's probably not so good. But science is driven by policy. Science has to be funded, and you can see in my presentation in Europe how much of science is driven by policy. And I'm so involved with research projects from EDCTP, from the European Commission, past with NIH and so forth, and it's really the budget for funding of science comes from the European Parliament and the European Council, and the Commission only exercises that budget. So it's really at the level of politics, as I think you're suggesting, that we decide about science. The second thing I want to put on the table is ethics. Ethics is new. I mean, it's like so new It's almost as new as gene editing, if you want. I mean, it's only since the 1960s that there's anything like an ethics committee or an IRB, right? It's only in the 1960s, maybe the 1950s, where the US, the Netherlands, and the UK started having ethics committees. Now, what, 60, 70 years later, we have an ethics committee in every country in the world. How is that possible? So ethics, the way we understand ethics, the way we talk about ethics today, is a new player on the block. And why did we struggle with ethics committees and IRBs, and why do we continue to struggle with them? And we do. In Europe, we struggle with them enormously. Okay? And why? Because we don't know what place they have in society. So we're trying to figure out what the place of ethics is in society. I think, with regard to your discussion, we have two things here. We have the ethics, we have the science, and we have this world of politics and policy that surrounds us, along with economics, of course, and things like this. I think that's why your meeting today in Bangkok is so important, and why we have to continue like this. We need to push for ethics. We need to insist on ethics, because ethics is the gateway. It will be the gateway for involving more people in the discussion. What's important to people with regard to their health? I think what's important, the most important thing for people with regard to their health is their decision. This is the most important thing for people. People want their voice to be heard, and especially when it comes to their own health, they want to be involved in their own decision-making processes. So, ethics is going to be a gateway, or it's going to be what, if we insist on this, then we can insist more on the engagement of persons, communities, and society in the discussion between science and politics, science and policy.
I totally appreciate what you're saying, and I totally agree. Being in Thailand, I just want to also offer that ethics is the foundation of Buddhist philosophy. So the ethics has existed at many, many levels, sociologically and theologically and culturally. For us to apply it to our medicine, we go back to the Hippocratic Oath, but really applying it on the edge of contemporary science is indeed a new effort.
Thank you. And I love to be in Thailand, because, you know, Thailand is, well, I shouldn't say it, but it's so Buddhist. I mean, it is so you don't even have to go to the temple. You're just surrounded by Buddhism, and the people, the culture, the society, the interactions. And of course, when you go into a Buddhist temple, it's the most amazing experience in Thailand. No other country has a Buddhist temple I know like Thailand does. So, thank you.
Thank you very much, Francis. We really appreciate you joining.
I told Daryl that he should invite two important people, and he managed to get one. It's Professor Suradej, who, as you have listened, he has been doing, interventions linking to genes modification. The other one is Professor Prasit, who is the chair of the working group on genetic technologies and its ethical implications. But I think Professor Prasit is not available, so I'm supposed to say something. But let me tell you, because I think what Franz has already mentioned about the European context prompted me to believe that what I'm going to share with you today might be another interesting addition to what Dr. Suradej has been talking, and why I will not be able to represent the view of the expert in Thailand about gene technology or gene editing. you might get some ideas to how we have been trying to grapple with the ethical issues in science and technology here in Thailand. As I mentioned to you, there's a body called the National Committee on Ethics in Science and Technology. And what do we do? Having listened to Daryl's introduction about ethics is an act of love, and here there are three types of ethics, descriptive, prescriptive, and interactive. This might be an interesting slide to explain a little bit more in detail, because when the decision was made to have a national committee on ethics in science and technology in Thailand, the question is, what do you want to do? What do you want to do? I'm a medical doctor like Dr. Suradej, and in medical professions, we have ethical compliance. Doctors will be so-called punished if they don't comply to the ethical rules and regulations or standards imposed within or introduced within a particular country. I used to be the subcommittee on ethical review. Of course, people file complaints, and you look at the behavior and you measure against some kind of so-called accepted standard. That's what we call ethical committee. I don't know whether it's really ethical in that sense or not. I personally believe, as Daryl has mentioned, that ethical concern raise dilemmas, and there's no easy solution. And as Franz has mentioned, it will not be easy, if not impossible, to try to say what is permissible or not permissible. So with that, we have been debating as to what the National Committee could or should be doing. This is what we have more or less concluded. Then the committee was established by the Ministry of Science and Technology, championed by a very prominent and leading scientist, Professor Yuttawong, who at one time used to be Deputy Prime Minister as well. He has been pushing for Thailand to have the National Committee on Ethics in Science and Technology, because he believed, as we have been talking throughout the morning, that science and society will always, interact, and at times came into dilemma, if not conflict. And the so-called the more advanced technologies seems to be tampered with normal human beings, if you know what we could mean by or define by normal human being, more and more, to the point that there will be definitely controversies and dilemmas. Having said that, he was also well aware that even though you use the term ethic, actually what we like to use here is what we copy from I think it was WHO or the American society when the Human Genome Project was very much active in the early 1990s, which is ethical, legal, and social implications of science and technology. So it's not only ethics that we are talking about here. It's what was called ELSI. So that's what we try to grapple with, which has a lot of so-called implications for the way the National Committee works. Because we were also expected to try to identify quite clearly what might be the legal and social implications, not only ethical. So, of course, the committee is not an executing body. It is more or less an advisory body. So we provide advice to the Ministry of Science and Technology on ethical, legal, and social implications and some of the possible interventions, as well as good practices. When intervention, it could mean law. Good practices could be, advices manuals, whatever you may call. And of course, the aim is to help to prevent, mitigate, or minimize the so-called undesirable consequences, whatever or however you define undesirable consequences. And as you may well know, this will help us bring in all the so-called ethical principles or whatever that we believe in. We also believe that although we know that it is not easy to reach conclusion or whatever, it will be important for the society at large to be informed. It might comply with what Daryl mentioned in one of his slides, that when we work on this kind of issues, the ultimate aim or what the best that we could achieve or should be aiming at is to make sure that we have a better informed society, so that people could be engaging in the debate in the best informed way as possible. Because this is not easy. There is no easy solution. Having been in a so-called policy making position, I also have to tell you that whatever dilemmas that is brought in, when it comes to decision, somebody has to make decision. That decision could be based on the debate in the society. It could be based on his or her own experiences. It could be based on the best evidences research, whoever brought in. It could be based on examples from outside. But someone has to make the decision. And he or she might be making decision regardless of what the society debate or talk about. But having a well-informed society which has a chance to debate and to discuss and share ideas would make him or her more accountable to his or her decision, rather than leave them at whatever it is and say, oh, it's not very easy, Yeah, he or she made the decision, so let's agree. Daryl mentioned briefly about one of the laws that we passed about surrogate mother. Actually, the law was proposed and passed while I was the minister. It was proposed while I was the minister, and I was the one who has to, listen to the proposal and accept it. There's some interesting story behind this law as well, in the sense that the cabinet is not the one who proposed this law to the parliament. It was members of the parliament who was prompted by some expert or some social activists who proposed the law. In the Thai system, you have to starting point, either the cabinet or the parliament. And when the parliament proposed the law, one of the cabinet members has to be listening to the proposal, accept it, and then accept or reject it. Of course, normally you accept it, and then you consider, and then give your feedback within 45 days. If not, then they will continue with that debate. And if there are good enough consensus, they could pass it into law without waiting for the so-called cabinet consensus. Anyway, I'm telling you this because even that law, which is, I would say, not that complex or controversial, took time, and there were lots of so-called different opinions. I'll spare you the detail, but just give you as an example of how we try to grapple with things that have ethical dilemma. Actually, it's not as complex as whether you modify life or not. But it's as complex as what the Daryl defined as one aspect of ethics, which is relationship. It tries to establish what kind of relationship is more desirable in terms of parenthood. If there are 7 possible parents, I'm not sure when we did that, but there was someone who said that in the future there are possible 7 types of parents. There could be now 10 or 12. What are the relationships between the various parties? Especially, what happened to the so-called pregnant women and the children? if the pregnant woman is not the same as the mother. Anyway, as I say, I'll spare you the details. For the time being, I mean, the National Committee has been convened for the last 4 or 5 years, and most of the things that they did was debating about contemporary issue. When I became the chair, I proposed that we identify some priority areas that we work on, rather than just debate within the committee. When I was managing the bioethics committee 10 years earlier, we also did that. We have planned to produce reports on what you could call advanced in science technology, but that was bioethics. This science technology in general. So we have 4 areas. One is, of course, advanced genetic and its related technology. The second is big data and artificial intelligence. The fourth one is what Ajahn Sorat is now leading, which is ensuring ethical research practices. And the fourth one is on climate change. And as I say, we emphasize on social communication. We also try to engage the younger generation. So there are certain projects that we bring in younger generation to talk about ethics. And interestingly enough, we have a project where the younger generation were asked to propose, and I think they proposed an activity which centered around a documentary called Future Baby. I believe that the producer or the director is Scandinavian, Ajahn Sorat, if you remember. Anyway, they have been organizing activities, show the film, bringing discussion about future baby. So reproductive technology and Thai society could be a very interesting topic area. We also try to establish international collaboration because, as in many difficult and complex subjects, having a good international partner would help the debate in the society be a bit more acceptable if not agreeable. In other words, you don't just bring in this kind of issue by not having a reference to the fact that the world is talking about it. You should not be ignoring that. So UNESCO has been one of the very active members. We used to work closely with WHO as well. If you remember, WHO produced a book called Genetics and World Health, where the leading author is Sir Professor Beryl, another hematologist who worked a lot on thalassemia as well. But anyway, if you are interested, you can go back to that one. There are certain issues and possible recommendations that we are now working on. As I said, the work group are still in progress, including Ajahn Sorat's work. I'll just share with you, just to give you some idea. On the genetics, originally we asked the working group to address the issue of gene editing, the very example that Ajahn Suradej has been presenting to us. But they decided that in terms of priority, gene editing may not be that controversial and easily available in the so-called controversial area as much as genetic testing. So they took on the topics of genetic testing. As I say, it could be that the committee is very practical. In other words, we find something that we need to act on rather than debated about. And genetic testing is now already available. Actually, there are a lot of so-called service units established. Many of them have been very much commercial oriented. And as you know, genetic test is very, I won't say vague, but it's not definite. If you listen carefully to Ajahn Suradej's presentation, he worked on thalassemia and he's continuing on culture diseases. Modifying genes affecting or resulting in what we call monogenic diseases is less controversial than doing anything with the so-called multigenic traits or characteristics. Many of the things that we talk about, intelligence, aesthetic, even heights, physical strength, they are all multigenic, meaning that whatever you did is not definite. Because these are the traits modified or determined by a number of genes, if you believe in genes, in the influence of genes. And you might not know enough. You might think that you know enough. Not to mention about genetic testing. Testing may make you aware of the presence of certain genes that could bring you risk, but they are all probability. How did you convey that kind of things? Because people have a very wrong conception about genetics and health, and they believe that whatever genes that you have will certainly be good or bad for you. So for the people it's 100%. For science it's X%. And the X is never 100, it's always less than 100. Anyway, so they propose, they are in the process of contemplating that, because genetic knowledge will be very complex. Gene technology, gene testing is not only about health, it's also about ancestral, about family ties and things like that. So they believe that we should not leave the so-called genetic testing only to the medical council, and we should create some kind of a Thai association on ethical legal certification and proper use of advanced genetic science and technology. That's what they are proposing. Of course, we are not too sure whether this will help to solve the problem. But apart from passing a law, which is very, very difficult, I'm very sure that you can debate about that law for eternity and never get it passed. So they thought that having a prominent group to bring in dialogue in the society could be a way to go about. They have some. Just to give you the information that when we work on bioethics, we have some pre-existing recommendations on ELSI, which has been distributed to research institutions, research funding agencies, medical council, Ministry of Public Health, as the so-called those who are supposed to act. We have certain recommendations already, but they are just recommendations. They are never law, not even manuals or guidelines. For example, we have recommendation on how to use fertilized embryo, on cloning, on genetic modification in germ cells. Something that Professor Suradej has already mentioned, so I won't be repeating that. Use of the so-called induced pluripotent stem cell therapy. The other thing that I like to tell you is that the medical council play a very important role in this so-called genetic technology application. They are also very concerned about the so-called ethical standards. They have a very, very straightforward and important concern, which is when should a new technology about genetics be made available and could be used or practiced by medical professions. It's very clear, very simple. They pass the rule. Again, as another so-called ethical standard to cope with all this new technology. That in terms of stem cell transplantation or use of stem cell, any method of use will not be approved by the medical council unless it has gone through systematic research, either abroad or in Thailand. In the Thai medicine technology regulation, there's a practice that no drugs or technology will be licensed for distribution unless they are licensed in the country of origin. So the criteria about systematically research and license in the original technology in the original country and then license in Thailand is a bit great. They are not the same thing. But anyway, I just wanted to let you know that whatever attempt you try to deal with technologies, you are always dealing with commercialization, industrial business practices, and they might not hold the same criteria or the same approaches to that. On gene editing research, as Dr. Suradej might have already mentioned, we don't have clear guidelines. We have Institutional Review Board, which is very conventional. They don't even consider a very complex issue like, I think it was Daniel who asked whether science comes first or ethics come first. Franz gives you some answer. I certainly agree with him. You cannot deter people's curiosity. You ask for the freedom to think. Scientists have the freedom to think and to act as a scientist. They don't care about ethics, although you may try to do something. Actually, let me share with you this one. Just on this point, that why the existing mechanism in ethics doesn't have very sophisticated conceptual framework. In the National Committee, we were introduced by Professor Yongyut, who is also a member of UNESCO, and he took part in an international meeting, and he showed us an example of research proposal review based on ethical ground that the European Commission is trying to use. It was very interesting. It's about 10-page long, and it asked the researchers as well as the research institution to answer a number of questions about how they anticipate or how they, identify the ethical implications of their research. I think that's a very interesting practice, but I'm very sure the scientists will always be against that, because many of the things that you will be explaining in the proposal will be very anticipatory. And once you anticipate that, or once you imagine about that, there could always be positive and negative consequences, and you cannot know for sure. And I always say, yeah, that's very correct. When you start to ask scientists to behave ethically, at least 50% will be up to who actually make use of that knowledge and technology. And I think we have countless of examples where seemingly important knowledge has been used in a very destructive way. But that just to tell you that when gene editing research proposal is reviewed by IRB, it would be mainly harm to the subject and nothing else. And as they say, if you don't have the so-called the criteria or the practices to allow you to talk about larger implications of the research, it may not be that difficult to get the proposal approved and complied. Anyway, of course, you could talk about the fact that if the subject is babies or adults, would it be the same? Not to mention about fertilized embryo, not even germ cells, fertilized embryo. And then of course you can have all kinds of gene modification of the germ cell, which is certainly not permitted. And then of course we have no clear objection on playing God. You mentioned about Thai being a Buddhist society. When I was working on bioethics, we try to listen around to Muslim, to Christian leaders, and to the Buddhist leader. It was very interesting. When we talk about, genetic modification, use of embryonic stem cells or whatever, the concern is more about killing rather than playing God. When do life begins, that we consider them as human and we are not supposed to kill them. It's about killing. It's about the right of life. It's not about modifying life. We don't talk much about modifying life or even creating life. In other words, we may not mind much about creating life. Anyway, just to give you some ideas to what we have been talking about. And of course, when we talk about ELSI, we always put them into the broader societal context. So many things need to be done beyond research conduct. I believe that as Daryl may have already shown in the Universal Declaration, one of the main targets of the Declaration is researchers and research institutions. But there are much more than that. I actually propose a very simple framework that whoever has power over the others would have to consider ethical criteria. And those who are in power consist of many groups. Researcher is definitely one of that group. Policymakers, of course. Professionals, of course. Commercial sector. There are so many groups in the society that has power over the others. And if they don't take into account what you could call ethical consideration, it might be a very, disorderly or whatever, confusing relationship. But anyway, so we try to look at the so-called system approach. We look at the product development, the product licensing, the product marketing, professional regulation, and that's how the group came to. The working group on genetic testing has tried to adopt and try to give recommendations as to what we should be doing with regards to all these groups, rather than just ethics in science and ethics of researchers. Yeah. Okay. Just my before last comment is that if you look at policymaker and societal concern in the ELSI in Thailand, I would say it is very low. It is very low. I think Professor Suradej has been doing courses for a long time, but not that many people are interested. Of course, you have to have clear and so-called concrete, undesirable consequences. Baby, the surrogate mother is one. It appeared in the press that the Thais were being abused by the Japanese. It's not about ethics, it's about sovereignty. Anyway, but the concern came in different forms. And it's also very difficult to communicate to the public to see the so-called the dilemma and the controversies. We are very much black and white society. We want quick answer, we want definite answer as to whether somebody is doing it right or wrong. And it's always, somebody said it's because the Thai is very much in the blame culture. We are very quick to blame others. So we always want to know whether something is right or wrong. Don't tell me that it's gray. We can't see gray. And then of course, as I mentioned, we talk about the fact that institution has also be responsible. So when Dr. Suradej was working on the ethics and researcher, we say that it might be interesting to talk about the responsibility of the research institutions, rather than the responsibility of the researcher themselves. The researcher could be held unethical, but the institute should also be held accountable for making or for enabling or for allowing or for whatever word that you may use, the researcher to have unethical conduct. So these are some of the things that we believe we might be able to bring in a bigger society. But let me close by sharing some of my ideas about the gene editing part that Ajahn Suradej has been talking. I believe let me start with Daryl's definition that ethics is an act of love, and curing disease is also an act of love. But medicine is not nowadays not only about curing diseases, and there are so-called different shades of disease severity. Thalassemia would be a good example. Then Professor Suradej raised the question of whether it's ethical to try to eradicate thalassemia. And he cites examples of countries who have succeeded it's not the same as eradicating, let's say, people who have the tendency to smoke, right? Or even people who have the tendency to have mental illnesses. For me, thalassemia is so obvious, a disease that we wish it went away. It would be unthinkable to wish that the mentally ill would go away from the world, from the earth, and to look for a world without the mentally handicapped. As I say, in one of the steps that we one of the time we work about the bioethics. In those days, we consulted some leaders in the Muslim country, in the Muslim religion, and I listened to the international deliberation from the Muslim people. One of the things that I learned, I don't know whether it's the same, but I never heard somebody say that in Buddhism, is that God created the disabled to make us feel love and empathy for others. In other words, without variety, without the disabled, if you may use that word, you may not be able to feel love. You may just think that everybody should and could be as good as the others. Anyway, I could be going for too far, but my point is that there are certain diseases that they might not be controversial, even though it applies the same technologies. But if you apply the same technology to other conditions, it might not be acceptable. So I always believe that technology itself, unless it has a very clear purposes, depending on what you mean by technology, has a purpose, and if you apply it for so-called better good, better health in general could be one of the examples. Getting rid of undesirable or suffering diseases would be another example. When Dr. Suradej was talking, he compared two technologies, two possibilities. A person born with thalassemia, which has to undergo blood transfusion, chelation, and live for another, let's say, 30 years, with lots of suffering time. And you have a technology that could help to get rid of that suffering, let him live longer. That technology has to do with gene manipulation. And you say that no, people could not manipulate gene because that's violating God's realm. I think it's very, very difficult to understand for me as a Buddhist. And I'm not too sure whether it's less or clearer among the Christian or whoever who believe in the ultimate gods or not. But let me finish there. Thank you very much for allowing me to listen to the presentation and sharing with you some of these work and thoughts. Thank you.
Thank you very much, Somsak. And in fact, we have a presentation from John that's going to be next after our photo. We take a photo before you have to leave us as well. But on playing God. So we'll see if it's really playing God. Carla has her hand up. And God has his hand up too, I think. Or her. Go ahead.
Pardon me, or her hand up.
Yes.
Excuse me. So I really appreciated a lot of your conversation. Thank you so much. I just wanted, from the Judeo-Christian Muslim perspective, so Tay-Sachs disease is certainly something within the Orthodox Jewish community where there was this big question of do you intervene with the whole genetic process, and it happened finally. One of the Orthodox rabbis' children were having Tay-Sachs and dying at a very young age, and what they eventually did was create a gene bank of the Orthodox Jewish community where Tay-Sachs is prevalent. And then the young people, when they're going to date, they'll tell them, you both carry this, don't continue, or you're going to have to use IVF when you have your children to get tested. And one of the very prevalent rabbis from in bioethics, really the one who's at the top of the field in bioethics, his son said, well, we want to try just doing it normally. And they had Tay-Sachs children. The second bound of children, they said, all right, we're going to do IVF. We'll do testing. So because we see the suffering that's happening in our own children and we're suffering from it, so they decided, in the case of Tay-Sachs, that they should do that. That's from the Orthodox Jewish.
Thank you, thank you. I have another good reference now for this.
Yes, Ryan, please.
Thank you, Doctor, for your presentation. My concern is what about the impact of politicians and sometimes their lack of political will and the wrong decisions in terms of, say, the implementation of research policies? What would you suggest in order to mitigate or even eliminate such type of wrong that can be committed against science and the work of scientists?
Let me just respond very quickly. I always believe in well-informed and active society, and in this particular case, because it has something to do with science, I think academic institutions have to be held accountable also. In other words, they should be also a very active players in communicating properly to the society, if not to the politicians. If there is anything wrong, they should be the first group to come up and then point it out to the people, or even talk to the people in power directly. I think these are something quite serious. If there is anything that, as you say, are the result of bad policy making because of ignorance, bias, personal benefits, and not so-called applying what you could call good science and good ethics. Actually, in Thailand, again, back to the point that we have been trying to promote evidence-based policy making, we have tried to call for what you can call engaged academicians. You are not just work to publish or to sell your science into technology. You are supposed to be also responsible for policy development and policy decision as well.
Daniel, please.
I have two questions, but first I wanted to say that I know Professor Somsak for about 10 years now, and he's being constantly credited as being the best boss ever by my close friend. Now for my two questions. You were talking about the freedom to think in terms of freedom of research, but I wanted to suggest that the field of research is being now full with incentives called patents. So maybe if you could comment on this. And also following Kala's remark, before my first daughter was born, I had this test, and it turned out that I had the gene of one of these Jewish diseases, genetic diseases, and it didn't matter because my ex-partner was from India, so I mean, no problem with this gene. But I was deeply disturbed by the language in which I got the letter from the Ministry of Health, because they said that I was the carrier of this gene. And until now, I don't understand how come I'm a carrier of this gene and not this gene part of myself, with all sorts of consequences, perhaps if we're not trying to bring a child, two people carrying the same, the other word, having the same gene, so I am this whole person with this gene. So if you could comment about these two questions.
Thank you for both questions. I think the second question is definitely something that is very true. In other words, science is not propelled by curiosity. anymore. It was also driven by profit, if not greed. I had one assumption, Daniel, to share with all of us here. I believe that good scientists are always driven by curiosity. It's just like good doctors are being driven by love for others and trying to cure the diseases. The fact that they have to be working in an environment which also reward them or pull them to try to have more so-called material or wealth gain is something that has to be dealt with by whoever managing the so-called working environment, the policy makers, the managers of the hospitals, or even their own colleagues. Having said that, I still think, as we say, that I think also France has said that if you are talking about science in general, without going to the motive behind science, there is no way that you can stop them or you can prevent them from going ahead. Ethics is never going to be a deterrent force for that. But we need to have maybe, if I may call that, some kind of vigilant mechanisms to monitor. In health we have the term surveillance, and I believe that something like that should exist in a situation where things could always go wrong because of the wrong motives or whatsoever. But criticizing, unless it is very obvious, criticizing and I think they're not going to be that obvious, criticizing or trying to prevent something from happening, because it has been very clear that it is driven by motives and not by to intend for others, could be a very difficult situation to do. And that's why having working in ethics in science technology for these past years, I always kept reminded that yes, it's the same as democracy, it's the same as good politics. You need vigilance, you need active citizens. The more difficult task is that you have to understand science as well. You have to understand science in depth. You should not be just science fiction oriented too much. Although it helps, but you need to understand the real science and then bring the debate in a much more so-called constructive way. But the second point is very, and I felt bad for that because we have been using thalassemia gene carrier here as well. And when you mentioned that, we have been very insensitive to use certain terms, and I certainly hope that the medical professions or whatever, the health authority, try to find some better way. I may use this as an example to think and see if we can find new terms, at least in the Thai context. Thank you.
Thank you. Other comments or questions? Okay, well, thank you very much, Somsak. It's most illuminating.